Welcome to this new season of the SDR Kitchen. I'm Fernando Larcher, and I'm hosting this episode together with my colleague Ellen van de Bogaard. And for this episode, which belongs to the recipe book of episodes, we have the pleasure of having Dr. Joanna Jackow. Dr. Jackow is a senior lecturer of molecular dermatology at St. John's Institute of Dermatology that belongs to the King's College London. And Joanna had become a faculty member of this in this institute after two postdocs in prestigious institutes in Paris and New York. And she will be talking today about genome editing in dermatology. That is her focus now in her lab as she's trying to develop new tools and new therapeutic modalities for genetic diseases, skin genetic diseases. So it's really a pleasure of having you, Joanna, here. And I will leave Joanna to share her screen for the presentation. I just have to say that after her talk, we will have people can use the Q&A button in the Zoom presentation to have questions. So we will get these questions and pass them to Joanna for discussion after these more or less 20 minutes of talk. So Joanna, please.
Thank you very much, Fernando, for the kind introduction. I will share my screen now. Can you see my slides? Can you see?
Yes.
Yes. Yes.
Hello, everyone. And again, Fernando, we know each other for many years now, and I've been, as I was PhD student and a postdoc, I was very inspired by his determination and developing and proving a different concept of gene therapy. And it's nice to have him here in this area and introducing me, and I'm very pleased to present you what I'm trying to do with my group at St. John's in London. So I will start with this question: is there a good recipe book for gene editing in dermatology? And you can see a list of our publication, but this list is really not ending. There's more in the field as well that's trying to write one, and we trying to find the best tools, the emerging technologies together, find the right combination of concept for delivering this tool into the skin, to the cells. And today, I hope in this talk, in the 20 minutes, I'm going to present you some concepts, some ideas, and where we're going with this technology in dermatology and in skin research. So why skin is a perfect kitchen for gene editing? And this is, maybe we can summarize, because skin is accessible, is renewable, and is compartmentalized in organ in different layers, so it's actually quite easy to access. There's a high burden of monogenic skin diseases that need emerging new technologies such as gene editing. What is also very important that the visibility of the phenotype is clear a clinical endpoint. So if we can see, if we can see the improvement immediately, if this is the healing wound or not appearing a blister of the skin. Also, the manipulation can be ex vivo or in vivo, and this is a huge advantage. And we have multiple examples of establishing this concept now in epidermolysis bullosa, and I will describe one as well. So what has gene editing to offer in dermatology therapeutically? And we can define this in few points. We can reverse directly the pathogenic variant at the genomic level, and this is different to if we introduced a copy of the exogenous gene. The correction is permanent if it's successful and can restore actually the expressed missing protein to physiological level. And we can target, and this is the whole aim of permanent therapies, the epidermal stem cells, and they have a clonal expansion. And if this is reached, then this therapy can be a permanent. And there is a lower risk of adverse immune reaction, and we can focus now on local delivery, which can then reduce the systemic risk. And this can be, for example, the first step of bringing this therapeutical product into the clinic. So how we can use this gene correction and gene editing in skin diseases? We can simply look at this graph here, and I will introduce all the systems in a moment, but we can have the tools, the editor tools, and we can then divide this in developing two strategies, in vivo or ex vivo. If we think of in vivo, then we can really envisage to deliver them into the skin directly via different vehicles and type of delivery mode, and then establish in vivo therapy. Or we can also use them ex vivo in cells that has been cultured, taken from patients that then would be edited and then proved for safety and then reintroduced into the patients by injection or grafts or different modalities. And as I mentioned before, there's a high burden of monogenic skin diseases that need urgent and new novel therapies that would provide a therapy, permanent therapy, and this is the list, and of a lot of them, and mainly here epidermolysis bullosa, that is the umbrella for different types of them, with mutated genes here, ichthyosis type of genodermatosis, and more that are still really in the developing stages. And this is a list that shows we are working as a community on proof of concept study for gene editing in skin. And this list is really going beyond this. And we have a recent first publication on very successful prime editing in junctional epidermolysis bullosa. So I haven't found a more comprehensive table summarizing all the up-to-date gene editing attempts, but yeah, that will probably be updated very soon. So now coming to what is gene editing and how we can define it. So, and because it's a series of recipe book, I will try to introduce it to you as a recipe book. So the most important ingredients for gene editing is the main component, which is the nuclease. And this can be in different forms. And this is operating on the DNA. And then to make it to work, we need some spices, and that is tasty, and also targeting the right position we want. And in this case, we need a guided RNA, which is a sequence that we can define, and this sequence is aligned with the targeted sequence we wanted to manipulate. And these ingredients are extremely wide now. We have a lot of tools, and I'm here showing you the most commonly used at the moment, and they can be presented like this: CRISPR-Cas9 base editors and prime editors, with the specificities that the CRISPR-Cas9 system are basically modifying the DNA with in different forms, and can either inactivate the gene or can correct the gene. And then we have prime editors that are correcting and changing some point bases in the DNA, while prime editors working with different mechanisms can even correct and change some insertion and deletion. So the main differences in these three systems is that the CRISPR-Cas9, the first identified and defined system for genome editing, is operating on double-stranded breaks of the DNA. And briefly, if it's only cleaving the DNA, then the DNA will be basically repairing the DNA without any particular sense, and this can only lead to a knockout of the gene or on not disactivating the allele. And then if we're providing a donor template in which we can incorporate our intended change we wanted to install in case of correction, then we can then prompt the DNA to homology-directed repair, and this can be used therapeutically. So now we have the new systems that has been introduced already some times ago, which are working only on a single strand DNA with single strand DNA breaks. And as mentioned, the base editors can nick the DNA and then make point mutation, and then the prime editing can nick, but then can really reinstall insertion, deletion, and the wider range of the pathogenic variants in the DNA. So these two systems, I will be talking a little bit more today, operating with two different mechanisms. And this is important to know from my talk that the base editors are working with a deamination process, and we can either have adenine base editors or cytosine base editor, depends on how the aminase is attacking the nucleotide. And for example, in case of adenine base editor, we can correct C to T or A to G mutation. In case of prime editors, we working with reverse. There's a mechanism that install the modification underlying the reverse transcriptase process, and we're correcting variety of mutation and I mentioned before. And so when we compare and contrasting these two systems, they have been proven to work. In case of base editing, it's very efficient system, and we can, it's been a variety of examples now in the literature and in my group showing that this system works if appropriated mutation sequences surrounding in the DNA, then it works very well. We have 70% of mutations in dystrophic epidermolysis bullosa that can be targeted with this system. Lower chance of indels mutation is a lower effect of targets comparing to CRISPR-Cas9. And the most important, I think very important for me as well, is that it works very well in non-proliferative cells or less proliferative cells. And the main limitation is the limiting editing window, which basically can prohibit to develop the starter sequence, the guide RNA that is important to go to the right position to make the modification. On the other hand, with prime editing, we can target all mutations. It's a more flexible system because we don't have any limitation with any editing window. There is a bit lower off-target effects than base editing, and this is the main reason why we also are quite interested in establishing this. And unfortunately, it's still less efficient than all the base editor types. Now, coming to a little bit prep work in our kitchen, how to choose the right editing strategy for skin diseases. And just briefly to show you, dystrophic epidermolysis bullosa is a disease I've been spending the last decade during my training and now in my own lab to investigating the pathomechanism of this disease and developing in vitro models, and also establishing and then trying testing the different genome editing system. And it's very devastating disease, as you can see on these pictures. Right after the birth, the blisters are visible, and they're converting in wounds, non-healing wounds, and chronic wounds later with the adulthood. And also there is a very, very high risk, especially for the severe affected patients, to develop squamous cell carcinoma. As soon as we can intervene and provide a treatment, which the gene therapy treatment can provide and prevent all the down the line consequences, and that I think is the most powerful strategy, thinking also of the young children. So collagen 7 is a large gene encoding 100 118 exons, and we have a variety of mutation that had been detected and published, including nonsense mutation, deletion, frameshift mutations, and all the variety of this pathogenic variant that has been described provide us a very good foundation to as a gene model to test all this technological system from genome editing. So pathophysiologically, in this disease, the pathogenic variants in collagen 7 leading to loss of type 7 collagen in the skin. And when the protein is not produced, then we have defect in adhesion between epidermis and the dermis because of the not present anchoring fibrils. As you can see here, this is normal skin producing collagen 7 and having this anchors. But if we have a mutation that leads to absence of type 7 collagen, that whole structures are missing, and therefore that leads to blister and the consequences leading to this severe phenotype. So starting our cooking lesson now, how do we edit collagen 7 gene for dystrophic epidermolysis bullosa? And this is the whole portfolio of systems and types of editor and the forms of editors, delivery systems, cell types, and finally the therapy that we have to think about. And we have to really assess this properly to beginning when we start to think of working on gene editing in the skin. And I will talk today about dystrophic epidermolysis bullosa and show you an examples of how we and why we decided to go with base and prime editors, why we decided to use the mRNA as a form of editors to introduce to cells, and why we decided for lipid nanoparticles and the cell types as fibroblasts and keratinocytes for now, and with the vision that we could, in a first instance, for example, have some type of topical treatment for this disorder. And I divided my the results part in the preparation work, safety, plating, and how can we do some test batches proving that these strategies are safe. So preparation work, we thought, okay, if we want to have a topical treatment for EB using genome editing, it has to be something that can effectively edit the somatic cells, the non-proliferating cells. And therefore, the first thought was, let's try base editing, because it has been described that it works in non-proliferating cells, such keratinocytes and fibroblasts, quite effectively. So we identified a cohort of patients harboring this C2T mutation, frequent mutation that occurs in some few patients in the UK, in London. And we cultured the cells from patients' biopsies. We characterized the cells, and we could demonstrate, because the patients that we work with the cells have heterozygote mutation, that type 7 collagen was the expression of type 7 collagen was reduced, but not completely absent. And the thought was, let's culture the cells and try to introduce first to see how efficient the base editing is in a bulk population of cells. And we use the electroporation system to test this first in vitro. And the decision for starting with the base editing is, as I mentioned before, because there's a lot of mutation, the pathogenic variant that is SNP and can be targeted by base editors. And that was our main decision for starting with this system. And at the time when we started this test and this project, there was a few base editors that have been developed, but particularly the EB8 system have turned out to be very efficient compared to the previous version, and that's why we decided to start testing this in our cells. And you can see here, this is the EB patient's unedited history sequence from Sanger sequencing, showing the heterozygosity of this mutation, showing here the double peaks. And after we applied high concentrations of this base editor, we could definitely clearly reverse and this mutation to 100%. And nonetheless, this efficiency and the this correction also leads to a bystander mutation, which is not uncommon in base editing. If we have a similar base that is really falling in the editing window, and it was the case with this cells. However, this bystander mutation was a silent mutation and was not deleterious for us at all. And we've been also checking other regions of collagen 7 if there's any other further modifications, but we could not find. So it was very successful experiment that really convinced us of powerfulness of this technology and of this editing strategy. And we then went further and thought a little bit about the safety, which is a very important point. And we assessed this globally on the genome level, first by using a predicted systems to identify 10 to 20 possible positions that could be very similar to our guide. And then we experimentally been testing this also using amplicon sequencing, and we couldn't find any modification as this level, which was very encouraging. We also assessed the mutation correction on transcriptome level, and we could show that the mutation was gone in transcript. And we also didn't see any activity of the deaminase on changing the A to Y changes in the transcriptome, which is a common process also in base editing. And further on now with another project, we also initialized first whole genome sequencing analysis, comparing the edited base edited fibroblasts with non-edited fibroblasts and aligning this to the human reference genome. And from our initial analysis, we didn't find any variants that would felt in the exonic region. So we conclude that there's no deleterious variant in the edited cells that could be detrimental for the cells, for the edited patient's fibroblast. So far with the technology that we applied to test the safety, we cannot report any adverse effect due to using this technology. Also, we've been very interested in testing if this base editing can also be applied on biallelic variants, and we've had cells that have had homozygote mutation, which was G2A, also very suitable for using adenine base editors. And we've been applying the same protocol and the same strategy in these cells as well. We ended up of correcting this up to 100%. So it's also with appropriated protocols, and if the design is suitable, the biallelic correction are also possible. As I mentioned, the prime editing has the advantage that can correct all mutations, and in collagen 7, we have the remaining 22% of other pathogenic variants are not targetable by base editing, so we still need to think of different possibilities. So it's good to have also some work done in this regard. And after several attempts and testing different systems that has been developed to date in for prime editing, we've been finally having a good results by correcting heterozygous deletion mutation in primary EB fibroblast. You can see here that's the normal sequence, and this is the fibroblast from patients having the deleted G here, and then after applying this really invented design of our prime editing guide, we restored that G on the position where it was missing. And to prove this further, we did Western blot of the cells, and you can see here the prime-edited fibroblast started to re-express and produce type 7 collagen that we could detect in supernatant as well as in lysates from these patients. So it's a good example of never give up if things are not working. And we also recently, from our experience, have written a little bit of compendium summarizing all the difficulties in prime editing in human cells and how to go about the practicalities. So if maybe some interesting notes can be found there. So, and now I would come to the part of delivering, how we plating this on the dish, what we have cooked, and start with some ideas that we are testing now in the lab. As you know, that different modalities, how we can deliver this using viral system, non-viral systems, but we've been quite interested as a group on lipid nanoparticles because of the inspiration from COVID vaccine, of the success of mRNA therapeutics that can be encapsulated in these lipids. And also because it's a simple and non-invasive system that we could combine and use as a topical treatment, and maybe also lowering the cost of the up-to-date reported viral strategies that are developed to deliver a gene therapy. And there's when you looked at the skin, there is some open doors for topical treatment, and we have assessed this just reading literatures and assessing some possibilities for us to go around. We can, of course, beside the topical delivery, if we have some good base editor system developed in lipid nanoparticle, we can always introduce this also through injections, intradermal or intraepidermal injections. So this is all the possible modes as well to explore. And the lipid nanoparticle as a platform for therapeutic gene editing, as I mentioned, is good because it's already clinically approved for mRNA. And this is also here the link to why we decided to work with the base editor in format of mRNA. And they are flexible and in design and can be easily scalable. They are quite highly reproducible and can be produced in GMP environment. And they have a larger packaging capabilities as well as fewer biosafety concern, but usually they're not transient expressed for very long time. And
Joanna,
yeah.
Considering time, we need to try to find a way to wrap up in order to leave some time for questions, okay?
Right. Okay. I will finish this very soon. So we tested these lipid nanoparticles in fibroblasts and using a base editor mRNA with a success, as you can see here, with correcting the mutation. And as I have to finish, I think with the take-home recipe and listen for dermatology is that skin is uniquely suited for gene editing therapies. And clinical translation is already happening using gene editing. And we really should not give up at this point. And the major challenges still remain the delivery safety at the scale bar. But this, with the experimental design around, it can be answered. And I'm sure that this in dermatology will bring some novel innovations. We have first test from kitchen to the clinic of approved therapeutics, not from the skin yet, but for sickle cell disease, but it's a product that is now really available to patients with a sickle cell disease and is a CRISPR-Cas based therapeutic product. And lastly, I just wanted to say that the chef specials in this addition is that the emerging gene editing treatment are on horizon. There's a recent publication from Stanford University where they used iPS corrected with CRISPR-Cas9 cells and produced some epithelial sheets, grafting on mice showing safety. And I think the next steps, according to this publication, is to initialize some first clinical trials based on this data. So I think the therapeutic gene editing for EB and other skin diseases will come. And then please, if you work on this topic, then continue. So thank you so much again for this invitation. And this is my group, my students, PhD students and postdoc that working with me and helping to bring this step by step further. Thank you very much.
Yeah, thank you very much, Joanna. We have time for a few questions, and I already see in the chat some questions coming up. One is about the time frame of the BE protein expression when transfecting your cells with the BE mRNA. So how long does the BE protein remain functional in the cells, and what does it mean for the risk of off-target effects over time?
Oh, that's a very good questions. I think from we haven't done any core study specifically to looking at the lifetime of the BE protein, but according to the editing experiments, we think the lifetime is up to 4 to 6 days.
Okay.
Of the mRNA.
Yeah. There's also questions about the topical administration. So how to make sure that you're delivering the correct amount to ensure complete or just enough genetic change. Would one type of delivery be preferred or multiple needed? And what if the skin barrier is not compromised? Would then you need injections, or would topical application still be a good option?
Oh, there are multiple questions, but I hope I can remember the first one. If
how to ensure the correct amount of delivery to reach the skin?
Yeah, so we don't have an answer to this question yet, but we I didn't have time to mention the properly. I only showed the slide, but we have a now mouse model that carries a pathogenic variant, and it's an ideal model that allows us now to do more precise testing. So we will move. I mean, we actually moved already on performing some tests, but we can then do dose escalation and see how much is needed to.
Yeah. And maybe also one question that I also had, I saw most of the results with fibroblasts, and the question also from one of the people in the audience is, are the fibroblasts really the right or the best target for this collagen 7 correction, or would basal keratinocytes be as good? And maybe the keratinocytes are less easily to edit. What is your experience with the difference between fibroblasts and keratinocytes?
Yeah, that's a great question too. Yeah, we started with fibroblasts because of the technical challenges that we had with keratinocytes and accessibility to the cells. And we, in parallel, now are starting to do also keratinocytes work. And in terms of type 7 collagen expression, yeah, both cells are expressing type 7 collagen. Ideally, would be good to target in vivo both cell types. Our lipid nanoparticles are not at the moment specific for any cell types. So, but we're working on another disease that is only related to keratinocytes. So all the questions that we hopefully can answer through editing keratinocytes in this type of the disease will be applied also to collagen 7.
Yeah, and then do you use primary keratinocytes or keratinocyte cell lines?
Yes, we do have access to primary keratinocytes, and for doing more gene editing experiments, of course, we need to immortalize the primary cells.
Okay.
To be able to perform more experiments.
Yeah. Maybe one final question is 2 questions about the experience with the LNPs. And one question is, what size are your LNPs? And what is the BE mRNA loading amount? So the construction of your lipid nanoparticles.
So I can answer the question on top of my head, how big are the lipids, but they in a size that they can go through the compromised skin and the construct. And the last part of the question was?
The mRNA loading amount, the BE.
Yeah, so depends on in cells. So we've tested different varieties, and I think on one of my slides there was a different dose escalation, but we've been successful with 400 nanogram and also with low amounts, and we also could show some concentration dependent editing effects.
And I guess maybe also good for the audience is that your email address is in this slide. So I guess if there's any questions, more specific ones, that you can reach Joanna on this email address. I don't see any other questions. Also, in due of time, I think we need to move on. And thank you very much, Joanna, for your great presentation and sort of highlighting the bits and pieces needed for this gene editing in dermatology. So thank you very much. And for everyone here and also others who will not be able to join us today, is that this webinar will be broadcasted open access on YouTube. So if you have any slides that you would want to revisit, the presentation by Joanna will be visible and will be accessible on the YouTube channel. Then we also would like to highlight the upcoming ESDR Kitchen episode is the Molecular Cuisine, where Nicolas Gaudenzio will present his work. He's a PI in Toulouse in the Infinity Institute and does really interesting work on how, for example, also mast cells and maternal stress affects the risk of atopic dermatitis. So if you're into this, please tune in on the 11th of February. And then we have a final slide before we close off, is that also we have the ESDR upcoming, always in September, but important dates for you now is that abstract submission will open soon at the 10th of February, and registration will also open on February 10. So meet us all in the beautiful Heidelberg, Germany. And I guess that closes this session.